So I'd like to introduce you all to Mo. Mo did his undergrad as a biology major at Indiana University, and then his PhD in ecology and evolution at the University of Chicago, working with Joe Thornton on evolutionary biochemistry. He was interested in recreating ancient genes and putting them into modern animals to test hypotheses about putatively adaptive evolving genes. Mo is currently a postdoc in our department with Trisha Whitkop here, studying basically how to understand how gene expression evolves and to complement his PhD training by working with proteins. Mo's research aims to understand the relationship between genotype and phenotype and the mechanisms through which that relationship changes as a function of chance and necessity during evolution to create the variation we observe. And today he's going to talk to us about genetic causes and organismal consequences of metabolic enzyme evolution.
Thank you, and thank you both for organizing this, and I'm sorry I got you some stuff later than I should have, but happy to be here. And it's really nice to see all of you. I feel like it's been a while since department with faces out and just, in a room together on a sunny day. So yeah, with that, I'll get started. And in today's talk, I'll mostly be talking about this thread of trying to understand evolutionary biology with a mechanistic and genetic approach. And I'll focus mostly on my PhD stuff, but as well as some of the newer stuff that I've been working on here in the Whitkop lab. But to begin, my research is motivated, like that of many other biologists, by variation. So some of the most remarkable features in the natural world are those that allow organisms to survive and thrive in different environments, that facilitate the fit of organism and environment. And evolution of these variations, whether they're adaptive or not, can happen at vastly different timescales. This can happen in the order of human lifetimes, as we've seen with the peppered moths, or across millions of years between species. These variants are all the products of a process of descent with modification. And many of us are evolutionary biologists because we want to understand how this process occurs. In my research specifically, I'm interested in understanding how this process occurs at the level of genetic changes and the molecular functions that encode. Kind of the explanatory framework I aspire to goes something like this. How do changes in genotype cause changes in phenotype? In turn, how does that cause changes in fitness of the organism in the environments that we think they function in? And of course, in time, how does that change the distribution of genotypes? This is how we think about the process of evolution. And of course, I'm not new in trying to understand evolution this way. This has been the framework for over 100 years now. What was fortuitous when I began my PhD is the challenges that existed prior. Many of them had been alleviated. They're not gone, they're still hard to do, but we can do experiments that we never could before. And most of my PhD work focused on trying to look at hypotheses of adaptation in this genotype, phenotype, fitness framework and use them to see whether the stories we told about evolution were true. And when they weren't true, what else might be happening? Of course, I don't work on birds or peppered moths or mice. And while I envy people who work on really pretty plants, I don't work on that either. But I would say I work on probably the most interesting system being portrayed here, which is here, an organism out of Africa with a weird propensity for alcohol. And I'm specifically interested in the role of the ADH enzyme in facilitating that adaptation of Drosophila and Monogastre to alcohol. One of the reasons I chose this system was because it was an iconic system in molecular evolution. So much work had been done describing variation at different levels, from the organism and its relationship to ethanol, to the population genetics, to putative features of the enzyme, that I could directly develop and test mechanistic hypotheses. So as a background on this system, the variation first became discovered in 1964, so predating the electrophoretic era that began 2 years later. Here you can just see, back in the day, I guess, you could run proteins on gels, and whether they were different or not was assessed by literally how quickly they migrated. Fast and slow were two of the first variants for evolutionary study. And ADH became interesting because it was clearly related to a trait that was important for the organism, ADH works in the first step of ethanol catabolism, turning over ethanol into acetaldehyde. And of course, Drosophila melanogaster was known for not only being able to tolerate ethanol, but actively seeking it out. Further, when the first DNA variation datasets came out, ADH was the focus. This was Marty Kreitman's work in 1983. And this wasn't just for the first description of population genetic variation, but numerous of the tests that he just called ADH the center of attention, and from an evolutionary perspective, no other gene has been studied as extensively in Drosophila as the alcohol dehydrogenase locus. So variation from all of these levels exist, and they allowed me to link together, or come up with a way of linking together how changes in genotype affected the enzyme fitness, and how that in turn perturbed genotype frequencies between populations and species. And it's kind of a matter of timing, because what happened in the literature is all of the narration of the hypothesis was kind of done by, I would say, the early 90s, but the precision needed to test that hypothesis really did not come about until later, or at least in a way that was feasible, for example, a PhD student to take on. But when I started my PhD in Joe Thornton's lab, I had access to the tools needed, and I could test these hypotheses, so I tested them. The first part of this talk will be looking at ADH variation at two levels. First, how did historical ADH evolution affect evolution of high ethanol tolerance in Drosophila melanogaster species relative to its closely related species? And secondly, why do different Drosophila melanogaster populations have different variants of ADH? So why is there segregating variation? And so in the first part, the structure of the hypothesis is this. Substitutions happened in the ADH enzyme, leading to the melanogaster lineage. These increased the rate of ethanol catabolism and in turn increased ethanol survivorship. The data to support this had been around for decades. So here, in some squish preps of melanogaster simulants and Drosophila ananasi, you can see that across life stages, the lysate has much higher ADH activity for melanogaster than they do for the other species. Similarly, if you compared melanogaster and simulants survival across different percentages of alcohol, you could see that melanogaster survives remarkably well, whereas simulants, which is very closely related to melanogaster, dies. Further, the first McDonald-Kreitman test found sequence patterns that were consistent with protein coding changes in the locus being adaptive. And before going on to what I did with the system, I just want to emphasize that many hypotheses of historical adaptation have similar structures to the ADH hypothesis. Change in some gene affecting some molecular function that affected some organismal phenotype. And what the approach I used of recreating ancestral ADHs and testing them alongside modern species, that approach can be used to test a lot of these hypotheses and shallow timescales, especially if you're working with model organisms, which still do have an ecology, even though they're model organisms. And in its simplest form, this is the hypothesis. There was some protein here, changes happened here, leading to functional differentiation of melanogaster, so blue and red should be different. And the way, so how do we do ancestral reconstruction? I'll very briefly go through this, but the premise is we can make a sequence alignment from extant modern day ADH genes. Once we've made that alignment, we can use maximum likelihood phylogenetics to infer the most likely phylogeny that explains them. And implicit in that phylogeny is a sequence that corresponds to each of these internal nodes. Once we know the identity of that sequence, we can then synthesize it. We can express it using an expression system, purify it, and we can characterize it alongside modern variants, just like we would with any other present-day genes. Of course, whether or not you can do this is completely determined by how much signal there is in your tree. So at every position, we can statistically look at the confidence we have in the reconstruction. The reason we can't do this with every gene or at every time scale is because your confidence just goes away as evolutionary signal erodes with time. So the quality control here is shown, which is for every site, we have basically perfect certainty of the inferred ancestral state. There's only one site where it's variable. It's this position right here, and you can see the reason it is because position 82 is toggling between these two states across the Drosophila phylogeny. All of the data I show you is robust to uncertainty at this position. I characterize both states. So with that in hand, I express ADH in a bacterial purification system. The reason for doing this is because that allowed me to get very precise quantities of protein that was unaltered in any other way. Why that's important is because there are two factors. There's both the function of the ADH enzyme, but also the quantity of ADH being made. And those two can have interesting relationships. So first, I just wanted to focus on the protein coding part because that's where the hypothesis was. Once I had these purified enzymes, I could just run them through a simple assay where we feed it ethanol with the cofactor NAD, and we could monitor the rate at which ethanol was converted into ethanol by looking at signal of NADH. We could do that across different concentrations of ethanol, and that would give us what's called Michaelis-Menten curves, but it allows us to infer parameters that explain the enzyme's behavior, the rate at which it turns over the reagent, as well as its propensity to bind to the reagent and so forth. So this is what the prediction would be. Ancestral ADH had low activity, Monogastre ADH had high activity. When we look at the data, okay, that is the property of the ancestral ADH, but the Monogastre ADH does not have higher activity. It is statistically indistinguishable in this assay from what we would have expected. Further, completely indistinguishable from that of the Drosophila simulans ADH. And simulans is not an ethanol tolerance. In fact, one of the big ecological differences between the two is Monogastre has safe habitats in basically vineyards and highly fermenting environments that simulants can't exploit. And this isn't a feature of our assay being not sensitive enough because we spiked in an enzyme that we knew had higher activity, and we can't pick up. So if we just look at parameters inferred from there, both the catalytic turnover rate, which is per second, how many molecules does a unit of the ADH turnover, we see that these three genotypes are statistically indistinguishable, as well as the affinity of the enzyme for the molecules, which describes the enzyme's behavior under subsaturating concentrations of ethanol. That too is indistinguishable. And for both of those, we can detect differences. It is not a matter of our resolution, at least with respect to about 15, 20%. But I told you that I express these proteins in bacteria, and in trying to get to a really clean system, did I reduce away meaningful variation? So do ADH enzymes behave differently when they're produced in flies? At the end of the day, this is what I wanted to do in my PhD, is do the stories we tell with test tubes, do they hold up when we introduce the organism? So what I did was I worked with David Lolan, who's now a professor at Williams College, and had also become fascinated with ADH around this time. And as he worked on the regulatory side, I worked on the protein side, but we made a common panel of genotypes. We took an ADH null strain and we integrated different, basically different alleles of ADH that varied only in their protein coding sequence. So unlike previous experiments, the regulatory variants that co-segregate with different species proteins were left out. So the only thing that were different were amino acid changes. And we created those flies and then we characterized them. First, we did what is kind of the organismal equivalent of the biochemical assays, but it's you squish up the flies and you separate out the soluble fraction, you quantify total protein, and you measure activity, scaling by the amount of total protein. And these assays results from these assays mirrored the results we had from earlier. That is, the ancestral ADH, the Monogastre, and the simulans variants all had statistically indistinguishable activity in the organism, or when produced in the organism. So we tested this part of the hypothesis and we found no evidence. But it is possible that ADH worked through other mechanisms to affect ethanol tolerance of plants. That it would be unintuitive, but molecules do weird things. So to test this, I took those flies with the modern day alleles in my little ancestral allele, and I went to Nebraska for a month and lived there and worked with my undergrad advisor, Christy Montu. And we basically brute forced tons of ethanol tolerance assays. One of the things I really wanted to make sure was I measured the phenotype at the life stage that I thought it was most important, and that was in larvae. The reason I say that is an adult fly can choose its environment in a way larvae can't. If a fly lands somewhere and it's too alcoholic, it can literally fly away. A larvae has to exist in that medium, eat what's there, and try to close. These were painful assays because you have to stage them at second instar before the larvae has already gotten big enough to just go pupate and take its chances. And we took replicate pools of 25 larvae. We placed them on tubes that had variable concentrations of ethanol that we had cooked in. And we just counted how many came out alive. That yielded these survivorship curves. And what you can kind of tell from there is 3 of these behave similarly and 1 of them does not. Of course, the 3 that behave similarly are the Monogastres, ancestral, and simulants alleles, whereas the one with our kind of positive control does survive better. I'll be kind of using data from these types of curves a lot, and I'll be showing you a summary statistic from them. That is the LD50, the concentration of ethanol at which 50% of the population is dead. And if we just look at that, it shows that we do have statistically significant differences. We also did this assay with adults, and what we found there was, at least with our experimental design, we could not find any differences, including the effect of the amino acid difference. So it turns out that was probably a good call to measure the larval level. But either way, those show us that, okay, the substitution of the Monogastre ADH did not change the rates of ethanol catabolism, nor did it affect ethanol survivorship. And we also went through and checked the population genetics, and kind of without going into the details of what happened, is the reason there was a signature of positive selection by the McDonald-Kreitman test on the ADH gene wasn't actually because of evolution in the Monogastre lineage per se. Many population genetic tests are whereas a hypothesis of adaptation on a lineage is not. What ended up happening was signal coming from other species, none of which have high ethanol tolerance, was causing that signature of positive selection. And that could be for a number of reasons, including variation in codon usage, as well as the fact that one of those lineages had a duplication of ADH, which fundamentally changes the constraint that exists on the protein and perturbs patterns. But so obviously negative results are not what anyone starting a PhD strives for, but all the way through, that is what I found in this case. But what I wanted to highlight was this was not a bad hypothesis. In fact, this was a really good hypothesis. The pieces of evidence were circumstantially consistent, just the mechanistic links weren't there. And when we tested them, we found no evidence. The bigger picture is of now we can recreate molecules that existed at specific time points in history and genetically trace that time through introducing the substitutions that we think are important and see what happens, not only in vitro, but also in vivo. So after that anticlimactic first half, I'm just like, okay, so why do different modern day Monogastre populations have different variants of ADH? And I will briefly just introduce that with a backstory. If you took populations of fruit flies from the tropics and you took populations of fruit flies from temperate or higher latitude regions, and you just gave them ethanol, they would generally classify into 2 different groups. The ones from the tropics have much lower ethanol tolerance, whereas flies from higher latitudes, and kind of amusingly, France actually have French fruit flies have the highest alcohol tolerance. They have very high alcohol tolerance. And kind of corresponding with that was variation in these 2 isoforms, or the 2 variants that were first discovered because of their gel migration speed. So they were called fast and slow. I'll call them K192 or T192. The reason I'll do that is because they also segregate with linked regulatory elements, and this keeps the fact that I'm talking about the protein coding change straight. So what else is interesting is not only is there variation in the trait, there is variation in the proportion of these enzymes. Just as ethanol tolerance increases in high latitude populations, the frequency of the T192 variant also increases. What is remarkable is this clinal distribution of ADH has been stable for the last 30 years. So this led to the hypothesis that selection was maintaining it. And there was a precise mechanistic justification for this hypothesis, or so it seemed, that there is a trade-off between enzyme activity and enzyme stability. One of the variants is just better at chewing through alcohol, but if it gets hotter, it can't maintain that activity and it just misfolds and degrades, so it becomes deleterious. Visually, that hypothesis looks something like this. T192 is faster at lower temperatures. As temperature increases, the amount of basically functional protein decreases, and it actually becomes relatively deleterious at hotter temperatures. Kind of alongside this mechanistic reasoning was the first applications of the HKA test and the coalescent with recombination. Both found, although the original papers were much more cautious about the interpretation than subsequent papers were, that the nucleotide patterns in the two variants were consistent with balancing selection maintaining it. And that, going back to the scaffold we had used earlier, looks something like this. There is genetic variation with the K192 mutation, and there is variation in ethanol tolerance. Presumably, the way they're linked is through the stability activity trade-off, where this change of increases activity, which is good, but decreases temperature stability, which is bad. And the balance of the good and the bad maintains the decline. This hypothesis is widely cited, but at the same time, it is one of those where, especially probably because it's with Drosophila populations, where we can see the gray data quite a bit better than we can with between species hypotheses, that there are a lot of pieces in the literature that are inconsistent with it. Nothing decisively about what is and what isn't, but different groups have found different things. And I thought this quote from Nick Barton from a Perspectives piece highlighted both the history of this polymorphism as a longstanding model, but also the difficulty of making unambiguous claims from the many things that have been reported in the literature. Nick said, even though ADH in Drosophila is one of the most intensively studied polymorphisms, we still do not know how its sequence variation has been shaped by selection. It is disconcerting that even the first and best studied example is still unresolved. And with the hubris of the youth I had at the time, I sought out to try to resolve it. And my first thing was by trying to reduce it to the most simple version, which is it is a hypothesis about an amino acid change. And does that amino acid change have the effects we think it does? The reason this was important is because all of the prior studies that had looked at these variants did not decouple the amino acid from the local elements that they segregated with. So even though the question is about the stars, the non-protein altering variants that are shown in little stripes here, they were there too. So the first part of this was testing these two predicted causal links. Does the mutation increase activity? Does it decrease stability? The first thing I did was take purified protein and measure if that was less stable, or if the two variants differed in their stability. The way I did that was with a method called circular dichroism. Basically, if you take folded protein and you blast it with polarized light, you'll get a spectral trace of it. ADH is a pretty helical protein, and the trace of it when everything is folded at a cozy temperature looks like this. As you start upping the temperature, the protein will start to melt, and this spectral trace will start to go away. From that, we can calculate its melting properties. So you can see that 20, 30, 40, the protein is fine. As you start ramping up, structure starts going away, and at a certain point, all of the structure is gone. So simply by looking at the signal at about 220 is where we look at for helical proteins, we can look at the proportion folded, and we can ask if this curve is different between the two genotypes. When we look at that, we again find that at least at the level of stability, there were no differences. Of course, we're looking at a pretty wide temperature range from 20 to 60 degrees. Fruit flies do not live at 60 degrees, so where would the meaningful information in this curve be if there was any? I have kind of benchmarked a few time points. So some strains start going into heat shock, males start becoming sterile around 29, 30 degrees. Around 34 degrees, the flies that I was working with were dead if you expose them there for 24 hours. And if you put them at 40 degrees for longer than 30 minutes, they were also dead. So even if there were some differences here, that is coming well beyond temperatures that would likely be physiologically relevant. But of course, that's the protein's melting properties. What about its activity? So to measure this, I basically took controlled quantities of enzymes and I assayed their activity at different temperatures to see if the T192 variant was more heat sensitive. So did it actually start losing activity as temperature increased, whereas the K192 variant would maintain its activity across the temperatures. And we see that there is no evidence for that. T192 is faster at colder temperatures, but both enzymes increase their activity as temperature increases, as one would expect. And at no point is there a switch in the rank order of the enzymes. So our prediction going in was this. Instead, we found this, that this new variant increases activity without decreasing stability. There is not a trade-off. But is there a trade-off at the level of the organism? And again, I first decoupled the effect of the amino acid from the rest of the gene. I used the same types of assays as I had before, and here I measured survivorship at 22 degrees as well as at 27 degrees. The reason I couldn't go higher was it was just very hard to maintain stable lines in the incubator above that. And 27 is around the upper end of the optimal temperature range of Monogastre. I looked at larval and adult life stages, and I won't be showing these data because they basically say the same point. I looked at chronic heat shock, I looked at acute heat shocks, I looked at sex-specific effects. The same pattern holds across all of that. So first, is there a temperature-associated effect? If we just look at them at larvae at 22 degrees, we can't detect an effect of the amino acid. But if we look at it at 27 degrees, we can. And it's in the opposite direction predicted by the trade-off hypothesis. The higher activity enzyme just provides higher ethanol tolerance without a seeming trade-off. And when we looked at adults, we could not detect any effect at all. One thing I do want you to notice here is there is an effect of temperature, which is the same points on this side are lower than the same points at that side. And I think that's important to keep in mind as we're breaking kind of one model of how ethanol tolerance variation happens, that we can think of other things that might be contributing to it. Because temperature in itself, so ethanol can be a dietary resource and it can be a toxin. And where one transitions from resource to toxin is mediated by temperature. And we can talk about this more later, but the key reason is because of the effect both have on membranes. And before showing anything else, I do want to point out that these are just small numbers of plots on a slide, but over 7,000 fly larvae were handpicked with a brush for each of these plots. So I just need the validation for painful experiments. Okay, so I looked at the effect of the amino acid and found nothing, but did the associated variants, did they have anything to do with it? Were they one of the reasons why results in the literature were confounded? So I also had a variant where I took a natural T192 haplotype with everything it segregates with, cloned them into the exact same locus and did the exact same assays. And when we add that in, we see nothing really changes at 22 degrees. And at 27 degrees, the whole haplotype, the T192 plus associated, it does have higher survivorship than the K192 variant, but no more than is conferred by the amino acid alone. Those linked variants don't have an effect. In adults, though, there is a different story. By adding in those variants, we see that the ethanol tolerance of the flies goes up quite a bit. And this work has been teased apart in really beautiful detail by David Lowland. And of course, I have to walk the company line here and say, so the amino acid and associated cis-regulatory variants increase ethanol tolerance at multiple life stages. And the mechanism at play is different across the life stages, which I think is really cool. And it turns out that Monogastre actually has two different promoters, or two different transcriptional start sites, depending on which life stage they're at. In some species, this is resolved with a duplication. In Monogastre, it's ADH. It's resolved by somehow they just evolved two transcriptional start sites. So I care about cis regulatory changes too. So again, we can come back here and we can cross off one part, but we actually find compelling evidence for the other. This mutation affects a molecular phenotype, and that phenotype seems consistent with increasing ethanol tolerance in certain environments. Further, this kind of T192 is just beneficial, seems to jive with the geographical patterns we observe, because in the temperate populations, the T192 is found consistently at higher than 80%, and in some populations it is fixed, whereas it's virtually absent in the tropical populations. So an alternative hypothesis might be that T192 is just a new variant that is beneficial in presence of high ethanol and segregates neutrally otherwise. So the reason I kind of like this framework is because I went in with one hypothesis, I tested them, found new data to support parts of it and to take away other parts of it. And in doing that, I can come up with an alternative hypothesis that in itself can be tested in the future. Of course, I walked in kind of linking together what other people had said, and a lot of the work is actually the descriptive biology that gives you the structure of the variation that we then go in and try to explain. And in the same way, I kind of called out the faults in previous hypotheses. I want to call out what mine fails to explain that the previous one did. This hypothesis that I'm putting forward relies on ethanol as a selective pressure that varies. We have no evidence for that. And we don't have evidence against it either, but we just don't know how the selective pressure would vary. And the other part of it is I don't know if ethanol necessarily has to vary or if the fly's relationship to ethanol can vary. So do highly ethanol tolerant flies know they're highly ethanol tolerant, or is the behavior actually evolving separately from the physiological trait? And similarly, does this actually have anything to do with ethanol at all? Because at the end of the day, even though French flies can grow up in up to 13% alcohol for their whole life, which is insane, they normally don't have to. And it could be that as a byproduct of selection on cold tolerance, you develop robustness to alcohol. And at least at the level of plasticity, we see that. The same genotype, if we raise it at a colder temperature, has higher tolerance than that genotype raised at a hotter temperature. Regardless, in my PhD, I tested two textbook hypotheses developed from work in population genetics, physiological and ecological genetics that had predictions, and how they affected adaptation. In both cases, I was able to test the causal links because in large part, I had access to equipment and the tech existed. And kind of in working at that weird interface of molecular biology and evolution, I could test the arrows that kind of guide connections across biological levels. And in both cases, I found that evolution happened differently than predicted. I think just in these cases, individually and together, highlight the importance of clear testable predictions. It doesn't really matter, or I guess I made it to the next stage. So whatever the cost of negative results were, they weren't that prohibitive. But for the purposes of science, it doesn't really matter if the hypothesis is right, so long as it can yield new information. The process of testing it can yield new information. And also having a PI that supported me in doing this and doing something completely different from that system was very, very helpful. So if my PhD was the causes and consequences of enzyme evolution in this fly drinking a beer, I then moved into this amorphous yeast that exists here and came here to work with Trisha on the causes and consequences of enzyme regulatory evolution. And kind of, if I look at my PhD and narrow it to a question, it was how did genetic changes cause phenotypic changes? The project that I'll talk about today from my postdoc is on the other side of that. And I'm putting asterisks on the not, but how did genetic changes not cause phenotypic changes during evolution? And the phenotype I'll be focusing on is a step in glycolysis. So glycolysis behind photosynthesis, which allows us to get chemical energy on planet Earth, is the process through which that chemical energy is broken down so life can exist. It is very fundamental, and the enzyme that I'll be talking about TDH or GAPDH. I'll be using the TDH number, but they're the same stuff. Works in there. And what's weird is like most things can get away perfectly fine with one GAPDH. Like Candida albicans has one GAPDH. Humans have one GAPDH and we're doing fine. This dog that keeps telling me I'm doing fine probably has one GAPDH. But why does Cerevisiae have 3? Not only do they have 3, the TDH enzymes are highly constrained, highly expressed, and they're strangely conserved despite duplications. One of the things that's supposed to happen with duplications is that it relieves some constraints. So usually things tend to evolve or change genetically a bit more if they exist with a paralog than if they're the only copy. So TDH2 and TDH3, which will mostly be the duplicates I saw a full rescue, it would mean it was entirely regulatory, at least with respect to this phenotype. And a partial rescue would suggest a bit of both. What we see from the data is just expressing the other TDH proteins with the TDH3 promoter is sufficient to rescue the growth defect, at least during this phase of growth. And this kind of first supports that the TDH proteins are approximately, or at least in this assay, totally functionally equivalent for this phenotype, and that it is the TDH3 promoter that's responding to cellular signals to produce sufficient quantities of enzyme during exponential growth. So that kind of working between this descriptive work and trying to get back to my comfortable, safe hypothesis land, the hypothesis that I could come up with is that the TDH3 promoter responds to cellular signals to express TDH as necessary to meet growth demands. So if that's the model, then we can expect something, or we can incorporate that information into this model. There is some wild-type level of gene expression, and deleting TDH2 kind of pushes you that way, but fitness, or in this case growth, it's really not that different. Most biological systems, at least at the level of pathways, evolve this kind of robustness, and it might just not be detectable in our system. But if there's a larger perturbation, like by TDH3, it does actually deplete the gene's expression to where the fitness effects are detectable. And if we're already working from there, then this much of a deleterious boost from TDH2, we add them both up, that further exacerbates the phenotype. If that is true, and if the TDH3 promoter is the site in the genome where kind of the metabolic states information is being synthesized, then we would expect that the promoter would be trying to compensate for this deficiency. The TDH3 promoter would be getting more signals to be like, go, 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 we have a problem, we need more. We could test that. The way we could do that is by just putting in a second copy of the TDH3 promoter, not coupled to a protein, but to a reporter that, that is, in this case was YFP. We would expect that in wild type, that reporter would be expressing approximately at the same level the demand for the TDH3 was. However, in a deletion, the cell would sense that it did not have enough TDH protein. And the way it would try to compensate would be by upregulating this promoter, so producing more yellow stars. And we could test how this was happening by concurrently measuring both how the culture was growing, but also how the YFP signal in that culture was growing with time. And we could look at this ratio and whether that ratio was different in different genotypes. So first, looking at everything but genotypes that had TDH3 deleted, that ratio looks like this. So here is reaction time, and here is the YFP level in the culture scaled by the OD. The reason I'm scaling by the OD is having more cells will just create more signal. So we have to normalize out the YFP signal by the number of cells in that culture. Most of them have this kind of dynamic where it goes up, down, and then up again. What does TDH3 deletions look like? As predicted, the YFP levels are much higher in the strains that don't have TDH3, consistent with the promoter trying to upregulate in response to insufficient dosage. And similarly, just as this model would predict, that effect is even stronger with TDH3 and TDH2 both being deleted. And now, so this is where I will stop showing new data, but this leaves us with, okay, a model that seems to be working, but where kind of the intricacies of it, which are fundamentally interesting, have not been worked out. What's the signal? So something is telling TDH3's promoter that the cell is not okay and it needs to put out more. And like it just, we have no idea what it could be. Is it a direct regulator? Is it being mediated through other things? And if there's a signal, then what's the responder? What in the cis regulatory sequence, that is the TDH3 promoter, is mediating this? Is it through the same sites that actually just lead to endogenous expression levels of TDH3, or are there different sites that transcription factors go to in these times of duress? And of course, this can open the pathway for these, we've been focusing on the TDH3 promoter, but what are the dynamics between the different TDHs? At some point, do TDH2 or TDH1's promoters also try to alleviate the stress? And is that just because of shared regulation because of their history, or is that because of other reasons. And ultimately, where I want to push this to is this kind of co-evolutionary dance between the three TDHs. Is it unfolding in the same way in different species lineages or not? So do the TDHs from different species play nicely with each other? Anyway, just with the data in hand, so far, the model that seems to be best supported would be there's some kind of expressions of functionalization going on. The reason I say that is because there does not seem to be any new phenotype. Rather, it seems to be an asymmetric distribution of activity with the TDH promoter being the biggest. But without really any data to support it, I will speculate that as we push further on the system, we will find both protein subfunctionalization and molecular neofunctionalization. The reason I say that is first, these are not mutually exclusive, and oftentimes I would expect that the first part would have been expressions of just because it is easier to change regulation than it is to a protein. Once that removed constraints, proteins could neutrally accumulate things that would be consistent with this complementary loss of protein function thing. And if you neutrally and complementarily lose functions, what that does is that just buys genes time. And if you can stay in a genome long enough, it just increases the likelihood by chance mutation. And through neutral wirings or potential adaptation, you can pick up a new function. And I'm not talking about any of the protein work we've started or looking at other things, but the reason, some of the empirical stuff that makes me think we will find that is because we're starting to discover functions of TDH well beyond glycolysis. We have been able to replicate literature from either the food industry or from cancer cell lines that the GAPDHs or for us TDHs go and play an active role in the nucleus. The nucleus is not where enzymes are supposed to, or metabolic enzymes are supposed to go. And yet we can see that they do localize in the nucleus. What are they doing there? And has there been divergence between the variants they're in? We don't know. And at the same time, we have just recently been able to show that changing the TDH genotype affects how Saccharomyces cerevisiae interacts with other species. In both of these cases, all I want you to see is that there are a lot of colonies here, and there are not as many colonies here. All we've done is we have changed the TDH genotype. And the reason I think that might be consistent either before or as the angiosperms proliferated through Earth. The reason that matters is if this is a fermentation behavior, fermentation as a global process became possible after fleshy fruits started appearing. So this may have been one of the opportunities that was created by changes on the planet that allowed this evolution of fungicidal behavior through otherwise basic metabolic proteins. So instead of like big data conclusions, I'll end with some concluding questions about this part. That is, how has the interplay of regulatory and protein evolution affected the co-evolution of and functional innovation in core metabolic enzymes? And what have been the consequences on the biology of the organism, from direct activities in its nucleus to its interaction with other species? And I think even though these are really different systems, really different types of experiments, this is still the scaffold that to me holds it together and why I want to be an evolutionary geneticist. It's that in my PhD work, I looked at the causal links between variation integrated across decades of observational and descriptive work. And in my postdoc work, I'm trying to do both. I'm trying to characterize the variation and come up, so like the text and hypotheses about the mechanisms, which are the arrows. But the goal is the same. I want to understand how variation at different levels affect and are affected by each other, from nucleotide changes to molecular functions to organismal phenotypes, and then use that information to hopefully better understand the super big picture questions. So how are chance and necessity, constraint and contingency at the molecular level, how is that shaping the variation we see? And ultimately that produces this. So yeah, thanks for coming. Yes. Well, so first, if we looked, I'll say two things. First, on the fly side, important changes seem to have been on the protein level in the larvae, which might be the stage that matters. So there's that. And then the second part is the way I would defend that is because I don't think the two are separate at all. I think what a molecule does is a function of when, where, and how much of it exists. And when and where how much of a molecule exists matters if only the molecule is able to do the things its environment demands or makes possible, adjacent possible. So there is that part of it. And the second part is, I really think that all phases kind of are dependent on each other. So it's, even with these where you're in a single cell, regulation, changes in regulation can increase or relieve constraint. That can open the way for proteins to do different types of things, which are often made possible by very small numbers of changes. And because that happens, I think in turn, regulatory networks can then start changing. And the most clear evidence for that we see is in transcription factor, cofactor turnovers, such as in Sandy Johnson's work, where that is exactly what seems to be happening, is without any kind of functional change at the level of the organism, proteins are basically handing off things to each other, and they're doing it because just the context for them to interact is changing because of their regulation. I hope that answer doesn't get me fired.
Yeah, regarding the variation or EEB across the subpopulations, you know whether there's gene flow.
Yes, and I think that is almost certainly what I think is maintaining declines. There is extensive gene flow between Drosophila populations, and it's made more complicated because the two places where you see declines, it's on the east coast of North America and along, I believe, the east coast of Australia. Both of those areas are where founder events, basically the north side got colonized by European populations, the south side by African populations. And European populations are nearly fixed for one variant. The southern populations are nearly fixed for the other. And as kind of gene flow and migration has happened, that kind would be one of the predictions. Now, what has to be the consistent level of mixture every season to sustain that? That's a very hard question, and that's exactly the type of question who one of our faculty that'll be arriving, Gideon, works on. So yes, that substitution, and we used that because I knew I could measure a difference, and that whether my method was failing, that was the sanity check or the positive control.
So sort of the narrative of that will provide
Exactly. So there's the first round of ethanol adaptation. Monogastre is more tolerant than the other species. That does not have that variant at all, because that variant doesn't exist until much more recently. That's explained by regulatory changes and changes at other loci. We do not. And we also, and some part of it is like, how does Monogastre actually interact with them? To ask the next set of questions. I think that would be the work we would have to do first. So the TDH2 and TDH3 promoters have some binding sites for the same transcription factor. In terms of conservation, they're not really alignable in any meaningful sense. But for promoters that diverged about 100 million years ago, you wouldn't expect them to be alignable. But the question that's getting to is just like, where are they in kind of like regulatory space? Are these two sharing common regulators or not? And that's actually exactly the type of data we're looking at and collecting like now. Okay, I have kept you from sunshine long enough.